are never static, right? Like I'm moving as I'm talking to you. Everything is moving. Life itself is moving. So can we make these structures such that we have control over a fourth dimension, that is time? So can we make dynamic structures out of this? So to do this, we took inspiration from biology. So let's take a look at DNA as it appears in nature, that is in biology. So if I were to take the DNA that's found in a single cell, and if I were to stretch out this DNA, that would be about 6 feet in length. But all of this DNA fits into the nucleus of one cell that is approximately 6 micrometers in diameter. So the question is, how does this DNA, which is 6 feet in length, fit into such a tiny, tiny space in one single cell? And the answer is, you can think about this DNA like you think about a ball of wool. So it somehow wraps itself into a ball of DNA such that it can fit in the cell. So the way this happens is it interacts with proteins called histones, and these proteins have amino acid residues that are positively charged. So these proteins have multiple positive charges, and we talked about the structure of DNA on slide 1. So DNA has a negative charge because of the phosphate backbone. So as a result of this, the DNA can wound around the histone proteins and consequently get condensed and therefore fit into the cells. So inspired by this idea of DNA interacting with with proteins that have multiple positive charges, I hypothesized that what if we make our crystals and then we use cations that have multiple positive charges? Can we then use the interactions of these cations with DNA and then make crystals that would be spongy? Cations would interact with the DNA and then squish our crystal, and then if you were to take the cations out, then it would go back to its original state. Can we do this? So this is what we investigated, and this work was featured on the cover of Journal of the American Chemical Society earlier this year. So let me briefly explain what we did in this study. So to test whether this thing would work or not, to test this hypothesis, we synthesized 2 sets of nanoparticles that are identical in shape, size, and DNA, except the DNA is complementary to one another. So if you have nanoparticles that are identical, but they only have complementary DNA, that's the only difference, they are expected to form a body-centered cubic structure. So you can predict the structure based on simple radius ratio rules that you use to predict the structure of atomic crystals, for example. So how do we verify that what we have indeed is a body-centered cubic? So you can do this by small angle X-ray scattering. So you would get some data. So you would see some peaks in the data, and you can fit it to the theoretical model of where we expect the peaks to be. And from that, what we see is that the theoretical lines that you see match very closely with the red line, that's the experimental line that you see. But what's even better is we can simply look at these structures under an electron microscope. And what we expect